To generate a new calibration, first select the New Calibration button from the toolbar. This will enable the controls in the Sample Details and Calibration Configuration panels. Then set the initial calibration configuration to be used. In this example, the spectra acquired from 4 calibration samples, each with a different concentration of molybdenum in a steel matrix, will be used. Initially, the calibration type will be set to peak intensity ratio. This requires the specification of an analyte peak wavelength and a matrix peak wavelength. The analyte and matrix names have also been added for reference. For now, the option to remove outliers from the data will be disabled. This option will be discussed in a separate video. 10 spectra have been acquired from each calibration sample to be used in this example, and this value is set in the configuration. Libsoft just uses this value to check that the correct number of spectra are selected for each calibration sample, and to warn the user if a different number are selected. In this example, the concentration of molybdenum in each calibration sample is specified in parts per million, ppm, and so this is also set in the configuration. Note that the concentration units are used for display purposes only and do not affect the actual calibration. The peak tolerance specifies the wavelength tolerance to be used when identifying the maximum intensities of the analyte peak and matrix peak, and can generally be left at the default value. The data points in spectra generated using the EMU spectrometer are uniformly spaced in terms of wavelength, whereas those generated by the Avantis spectrometers are not. As a result, the processing of spectra from the EMU and Avantis spectrometers needs to be subtly different, and the type of spectrometer used to acquire the calibration spectra is therefore also specified. Finally, the regression type to be used to fit the calibration curve to the data can be specified. This can be either linear or quadratic. Note that the calibration configuration can be modified as required, either as the calibration sample data are loaded or when the initial calibration is complete. This allows the effect of changing parameters to be assessed and the calibration to be optimized. Once the calibration configuration has been set, the calibration sample data can be loaded. In the Sample Details panel, select the New Sample button. This displays a New button, allowing the spectra acquired for this sample to be selected. Click on this button to open a file browser and select the spectra for the new sample. In this example, the 10 spectra for each sample have been saved in a single file, but the spectra could be saved in separate files depending on the acquisition configuration used. Provided that the number of spectra selected matches the number specified in the calibration configuration, the selected spectra will be loaded and listed in the Sample Details panel. Note that the first of the listed spectra is displayed in the Current Spectrum panel, and the analyte peak and matrix peak for this spectrum are also displayed. The calibration sample name is automatically set to the root of the file name of the selected spectra, but can be edited as required. Enter the analyte concentration, which in this case is 0 ppm, and then click on the Save Sample button. The data for the new sample are then added to the calibration. The calibration metric is calculated and plotted in the Calibration Preview panel. Note that the error bars on each point correspond to plus and minus 1 standard deviation from the mean. The new sample is also added to the list of samples for the current calibration. To add data for further samples, simply repeat this process. Note that when sufficient samples have been added to the calibration, as determined by the specified regression type, the calibration equation is derived from the data and the corresponding curve is plotted in the calibration preview panel. So in this case it's a linear calibration and we have 2 data points so far, so we have a linear regression line. Add the other 2 points. Note that selecting a different spectrum from the list will update that displayed in the current spectrum panel and also the analyte and matrix peak panels. A different sample can be selected for display either by selecting directly from the drop-down menu or by cycling through the list of samples using the forwards and backwards arrows either side of the list. To change the data for a sample, select the Edit Sample button. Make whatever changes are required. So we might say this is 4000 ppm instead of 5000, and then press the Sample Save button again. And the calibration is automatically updated. We'll just change that back to the correct value. To remove a sample from the calibration, select the Delete Sample button. And again, the calibration is updated automatically. But we will put that sample back again. In the analyte and matrix peak panels, the peak wavelengths specified in the calibration configuration are marked by vertical red lines. If the sample is changed, the intensity of the analyte peak can be seen to also change, whereas that of the matrix peak is relatively constant. If the calibration type is changed, the calibration is automatically recalculated using the new metric, and the analyte and matrix peak displays are also changed accordingly. For example, if the calibration type is changed to peak area, the matrix peak panel is hidden, and instead of the analyte peak being marked, the analyte start and stop wavelengths specified in the configuration are marked with vertical green lines. These wavelengths define the bounds of the peak for which the peak area is calculated. If the calibration type is changed again, this time to peak area ratio, the matrix peak is redisplayed and marked with the matrix start and matrix stop wavelengths.